I'm an undergraduate student at University of Salerno, and this is the presentation about this project that we conducted some years ago that is about the exposure of the human body to ionizing radiation. In particular, I focused on the impact of this radiation on the changes in the DNA methylation levels inside cardiac cells and the possible link to cardiovascular diseases. So the space radiation in general is composed by different elements. We have some low dose radiation, then we have some low dose rate radiation and highlight particles. And in general, when we are talking about the space environment, there are some kind of energetic events like cosmic rays, solar particle events, and the geomagnetic fields that stores some kind of radiation and heavy ions. And in fact, you can see that we have different elements that make up this kind of space radiation. What is the biological impact on human cells and in general on the human body? So when we have cells that are irradiated with this kind of energy and radiation in literature, but also considering all the experiments that are ongoing now, we have seen lots of different responses. There can be apoptosis, for example. There have been evidence of necrosis, genetic mutations that lead to a genomic instability that is then linked to the onset of human cancers. There is cell senescence that can be very dangerous considering long-term space missions. And there can be also rearrangements of the structures of chromosomes. Also, moreover, we have other kind of results of this chronic irradiation. We have some effects of the acute exposure. We have some effects linked to the chronic exposure that also changes the dose that is absorbed by the human body, and also the bystander effect that is linked to the interaction between cells that are irradiated and cells that have not been irradiated yet. In general, when we're talking about radiation, we have different kind of units of measure. For example, in general, we talk about radioactivity with Becquerel and Curie. Then we have the absorbed dose, that is the energy deposited inside the tissues and organs in grays. Then we have the equivalent dose that depends on the kind of radiation. And then we have the effective dose that depends on the kind of tissue and its radiosensibility. So we conducted this research some years ago at the Blue Marble Space Institute of Science. And we have published a preprint last year, and we tried to model the space radiation on the human body with the help of a particular software and package. It is the Gent4, and basically we created a human phantom to mimic the exposure to space radiation on the human body. And then we compared the values of these doses in the different organs to the ones that we know in literature that come mostly from some nuclear accidents and patients that underwent radiotherapy. That were the only kind of comparable radiation doses. So, for example, for the art in specific for this presentation, we managed to get some results, and we calculated the effective dose for this organ that were around 1 and 2.5 sieverts for the male and almost 1 to for the female. So they're kind of comparable between each other. If we consider the current limitations, for example, we have this NASA report in June of 2021. In the whole career of an astronaut, we should have an exposure of maximum 0.6 sieverts. And I want to remind that these calculations were based on a long-term space mission to Mars. And so considering that we have an effective dose that is higher than the limitation, we can consider that there could be a serious health hazard linked to this mission, this kind of missions. So the ionizing radiation is well known for its genotoxic effect that is able to disrupt the helices of the DNA with a direct action or an indirect action with the formation of reactive oxygen species. But at the same time, we don't have to forget about the epigenetic changes that are mostly the DNA methylation, the histone modification with particular enzymes, and also the alteration of these non-coding RNA that are able to modulate the genic expression inside our cells. So for this presentation, I focused on these elements that are called line 1. And in general, inside our DNA, almost 45% is made of transposable elements, which are mainly constituted of DNA transposons, LTR retrotransposon, and non-LTR retrotransposons. And line 1 is this long interspersed element that is linked to retrotransposition, and it is estimated that it makes up to 17% of human DNA, and thanks to its particular cycle and its expression and translation in proteins, it was able to generate almost 1/3 of our genome, because it has 2 overreading frames, 1 and 2, and inside the 2 we have, for example, retrotranscriptase. So the first transcript is created as a whole, then we have the production of ORF1 and ORF2, and then the ORF2, thanks to the retrotranscriptase activity, is able to integrate other copies of these structure inside our DNA. So that's why it generated so much of our DNA. Why did I choose this one? Because line 1 makes up 70% of the genome, and along with the allele sequences, it makes up to the 30% of genome. And considering that more than 1/3 of all the DNA methylation happens inside these repetitive elements, lots of scientific literature is agreeing on considering the methylation of line 1 as a good surrogate for considering the global DNA methylation. So it is a good indicator for this one. And also from literature, we can derive that the hypomethylation of line 1, but in general, the disruption of the methylated sites is linked to different kind of human pathologies. So we have the onset of cancers, the onset of some psychiatric disorders, metabolic disorders, and what we are focusing today is cardiovascular disease. So I chose 2 interesting articles from literature that are mainly focused on line 1 methylation, and they made use of this particular line of mice, and they also implemented some radiation that is able to mimic radiation that is present in space. In this particular case, we have protons and heavy ions of iron. So you can see here that I took these 2 images that represent the results of a methylation-sensitive RT-PCR that are able to calculate the levels of methylation into the 2 open reading frames of the line 1, as I showed you earlier. And you can see that at 7 days and 90 days, there weren't significant changes, while here we have significant changes both at 7 days after irradiation and at 90 days after irradiation. In particular, here you can see that there is a sort of hypomethylation, while here we have an hypermethylation, both for protons and for the heavy ions of iron. At the same time, you can see here that other parameters were calculated. We have the expression of line 1, so considering the transcripts, and you can see that there is a reduction when it comes to the exposure to the to the ions of iron after 7 days. And here you have some elements that belong to the methylation machinery. So we have, for example, the NMT1 and 3A that are linked to the maintenance of methylation. And TET1 is an enzyme that is actually linked to a process of demethylation. And you can see that it is significantly decreased after 90 days of the irradiation, both for the protons and the 65 iron ions. So if we want to sum up all these results, you can see that there are some increase in the methylation after 90 days, basically, in terms of statistical significance. And at the same time, you can see that there is a decrease at first after 7 days, and then there is an increase of the methylation agents at 90-day time point. So what do these results tell us? Basically, that after the radiation at 7-day time point, so an early post-irradiation time, there is an evidence of an hypomethylation. And this is also supported by other studies that show an early overexpression of another agent. And at 90-day time point, so very late from the early radiation, you can see that there is a DNA hypermethylation, also because of the important decrease in the TET1 enzyme that I showed you earlier. Another interesting article that is foundable in literature considers the exposure of cardiomyocytes and in general to the earth to oxygen ions. And the procedure was kind of the same. So they chose the same line of mice, and in this case they chose radiation coming from these oxygen ions with these particular energies, so up to 1 gray. And here they studied the line 1 methylation levels, always with a particular real-time PCR. And they divided line 1 based on the evolutionary age, because they are well preserved during the evolutionary process. And you can see that at 14 and 90 days, here, for example, there is a significant hypermethylation, while here at 14 days, there is a significant hypomethylation in different kind of elements. So it's not general, but it's only specific for some kind of elements divided by their evolutionary age. And at the same time, they analyzed the same methylation agents that we saw earlier. And you can see here that there are some hypermethylation, some upregulation of expression, especially when it comes to the 14 days time point. And the effect is more evident for higher radiation doses rather than the lower radiation doses. And here also they studied the metabolism of these methylation processes. So they studied the redox status with the GSH and GSSG and their ratio, and also the quantity of cystathione and the methionine inside the heart cells. And here I summed up again all these results. So you can see that there is an hypermethylation in 90 days that is in agreement with the earlier article that I showed you, while at 14 days there is always this evidence of hypomethylation. So in general, from these articles, we can derive the idea that ionizing radiation is not only able to alter the genetic material with the disruption of the helix, with the formation of strand breaks, but also is able to alter the gene expression, working on the epigenetic machinery. And there is evidence from human studies of a relationship between the levels of DNA methylation and the onset of cardiovascular diseases. For example, it is known that DNA methylation is not only important for the pathological part and expression of cardiomyocytes, but it's also important for their development and maturation. So it's important during the embryogenesis. It's important for the differentiation and the working of the tissue, in this case, the heart, but it's also important for the disease. In fact, there were studies conducted on the human heart, taking samples of left ventricle tissues, and you can see that cardiomyocytes that are linked to heart failure tend to show a methylation pattern that is very similar to the fetal phase. So it kind of shows a reprogramming of this epigenetic process. And also these DMR, that are the sites that are differentially methylated, were strictly close to some kind of genes that are very important for the cardiomyocyte activity. In fact, they were linked to genes that were involved in the cardiac development, the muscular structure, to the morphogenesis, that is very important for the heart to contract and conduct the electric impulse, and the energy metabolism, considering that heart is one of the most important and one of the most needy of oxygen to work. And here it was the work that I cited here. And you can see that there is just a partial juxtaposition between the disease sites of differential methylation and the postnatal that are the normal ones. And the most of these disease DMRs belong to the intergenic elements. So this is in agreement with the finding in research that line 1 and, for example, satellite sequences can be linked to this kind of pathology when there is an abnormality in their methylation levels. And other findings, for example, show us that these satellite repetitive elements, if they are hypomethylated, as we saw at the 14 days time point in the other article that I showed you earlier, there is an increased transcription. And also there is evidence that people that have end-stage cardiovascular diseases have an hypermethylation of these repeat elements that belong to the centromeric region. And hypermethylation, if you remember well, is one of the markers that scientists have found at the late time point after the radiation with doses that mimic the space radiation. So there could be an alteration of DNA methylation after some kind of time during the space mission after the exposure to space radiation. And to sum up also this kind of evidence, so the end-stage cardiomyopathy is linked to this hypomethylation that also is linked to an increased transcription, because usually methylation silences the transcriptional process. And so there is this kind of upregulation that we also evidenced with the 14 days and 7 days time points in the earlier articles. And the hypermethylation, there is this suggestion of an involvement in the heart disease, but it's still unclear. So there should be more research to understand if the hypermethylation of these kind of repetitive elements could be linked to the heart failure. And if there could be the discovery of this correlation, we can also assume that the hypermethylation that we saw in mice after the exposure to space radiation, to the radiation that mimicked the space radiation, we can consider that the long-term space mission and the chronic exposure to radiation could be a risk, a healthy risk for the astronauts.
Roberto, if I could ask you for a few sentences of summary, because our time seems to be up. So if you could.
I think actually I concluded. So I'm done. So to conclude very quickly all these evidences, we need to keep studying the ionizing radiation doses for long-term space missions that can be very useful for the plannings to Mars and the lunar missions. There is a need for more evaluation for the DNA methylation levels and the specific DMR sites. And in order to countermeasure the possible onset of heart failure and other cardiovascular diseases, we need to understand if we can implement pharmacological rather kind of shielding strategies during these missions. So that's it, and thanks for the attention.